is Jacob Kaitner. I'm a member of the Myers Lab at the Huntsman Cancer Institute, and I am working on determining GRK2's role in the Hedgehog pathway. The Hedgehog pathway is a cell fate pathway that is heavily involved in tissue development and maintenance. The Hedgehog pathway, when it's in its active form, helps cells respond to external stimuli and differentiate appropriately. So while this pathway is incredibly important biologically, when it is overactive, it can lead to cancers such as basal cell carcinoma, and when it is underactive, it can lead to developmental disorders such as polydactyly. So to better understand and treat these diseases, we first have to understand the Hedgehog pathway at a molecular level. And the interaction I am interested in starts with smoothened, a 7-pass transmembrane protein that acts as a molecular switch at the membrane. And when smoothened is on, it leads to the activation of GLE, which promotes the transcription of genes that control cellular identity. And the mechanism that is not understood is the communication between smoothened and GLE. Our hypothesis seeks to understand that. So our hypothesis is based on the idea that GRK2 is essential for Hedgehog pathway activation, and that it is known that GRK2 can phosphorylate active sites on smoothened. So we propose that GRK2 phosphorylation of smoothened stimulates GLE activation. And we believe it does this through the binding of a downstream protein to the cytoplasmic tail of the phosphorylated smoothened. And this downstream protein then leads to the activation of GLE, which promotes transcription of Hedgehog target genes. So to test this hypothesis, we identified some new phosphorylation sites, and we did this through the purification of smoothened from cells that we treated with GRK2 inhibitors, and in comparison to cells we did not treat with GRK2 inhibitors. We digested these proteins and set them in for mass spectrometry, and mass spectrometry is sensitive enough that it can detect the difference between phosphorylated amino acids and non-phosphorylated amino acids. So in the past, in vitro analysis has shown certain phosphorylation sites. However, this in vitro analysis was done with only the cytoplasmic tail. We now know that this cannot go into the correct conformation state to be properly phosphorylated for the activation of the pathway. So we confirm this here with our analysis because we found new phosphorylation sites that are smoothened GRK2 activity dependent. To test these sites, we mutate them to alanine, a non-phosphorylatable amino acid. We then test the activity of these mutants in the GLE luciferase assay, an assay that utilizes GLE binding site to transcribe luciferase, which, when in the presence of substrate, emits a measurable light. We did this for both smoothened, wild type, and the alanine mutant we created, in the presence and absence of Hedgehog, the ligand that causes pathway activation. The results of this assay show that the smoothened alanine mutant has a significant reduction compared to the wild type smoothened. So I then tested this mutated alanine form of smoothened in zebrafish, and zebrafish have proteins that are dependent on Hedgehog pathway activation. And below we can see these proteins in pink and green. So in wild-type smoothened, when we inject this into smoothened knockout fish, we can see appropriate rescuing of zebrafish embryonic patterning. And in the alanine mutant, we do not see this because Hedgehog pathway activation is not sufficient to cause it. In conclusion, the mutations of smoothened phosphorylation sites exhibit reduced GLE activity in cell culture and improper embryonic patterning in zebrafish. So GRK2 phosphorylation of smoothened is necessary for pathway activation, and this is important because now we can use this to target Hedgehog-related cancers. In the future, I would like to identify what protein GRK2 phosphorylation stimulates the binding of. And additionally, I would like to know what other components between this unidentified protein and GLE are necessary for the activation of GLE and the transcription of Hedgehog target genes. I would like to thank the members of the Myers Lab for their support throughout this project. I would also like to thank Ruth Hootenheim for the mass spectrometric analysis. I would also like to thank David Grunwald and Dana Shaw for their help with the zebrafish project, and I would like to thank the UROP program for their funding of my project.